So Jared, the third agent we had approved in this space is atezolizumab, and it was really on the basis of a phase 2 Poplar trial as well as a phase 3 Oak trial. Could you just kind of briefly walk us through that data set?
Sure. So the first of the Poplar was a randomized phase 2, the Oak a randomized phase 3, both docetaxel versus atezolizumab. And both showed the exact same finding, a hazard ratio of 0.73 for overall survival. I guess Oak read Poplar before being reported. I'll try harder on the jokes in this session. But exactly the same result, very similar efficacy and toxicity profile to what we had seen before in the comparative studies with nivolumab and pembrolizumab.
So it's interesting that the three agents we have took slightly different approaches. Nivolumab, a histology-based approach with Checkmate 017 and 057 being squamous and non-squamous respectively. And with pembrolizumab, a PD-L1, a biomarker strategy. And with atezo, kind of an all-comer strategy, right?
Yeah.
Yeah, and all in the same trial. The issue is that atezo is the first anti-PD-L1. Does that make a difference versus anti-PD-1?
You know, there are theoretical reasons why it could and perhaps should. But if you look at the clinically available data in this context, as well as other cancers in context, there's really no hint that it actually does. The toxicity profile looks nearly like a copy-paste. The efficacy profile looks nearly like a copy-paste. So at least in terms of single-agent use, atezolizumab looks more similar than different than nivolumab and pembrolizumab. What I'm curious about is to see when our Lord knows how many combination trials we have going on, but to see in the combination trials with atezolizumab and durvalumab if that story continues to play through of similar efficacy and tolerability.
So everyone on the panel has used atezolizumab?
Yes.
In your experiences as a group?
Copy and paste.
Copy and paste?
Pepsi-Cola and Coca-Cola so far.
Okay.
Although I have one story of a patient who's been on atezolizumab on a trial, and we had to come off after being on it for about a year and a half because of he had a lesion that we wanted to radiate, and the study wouldn't allow it, so he came off. And to make a long story short, we had to put him back on another agent. Atezo wasn't approved, so I put him on nivolumab. And after about 2 treatments, I said, so we'll just call him Mr. Smith. So Mr. Smith, it's the same thing, right? And he says, no, it's very different, very different.
Really?
And so, you know, I don't know that we have that many patients who've been on different agents
Right.
who can tell differences. But, you know, my guess is that now that these drugs are on the market, and we have no data to say that you should do this, but there might be some switching like this that happens for various reasons, maybe an insurance reason or some other reason. And I'm wondering if there'll be differences patients notice.
I had the same scenario though, exactly, clinical trial, and the person said there was absolutely no difference. Other than having to come more often.
Yeah, right.
Well, that's the one difference here, right?
Yeah.
So there are practical differences in how these drugs are approved, even if a lot of us aren't convinced that they're more different than, say, Coke and Pepsi. Nivolumab is approved in the second line as an every 2-week drug, regardless of PD-L1 status. Pembro is every 3, requiring PD-L1 positivity, and atezo is every 3, regardless of biomarker result. And those do influence when you can prescribe them vis-a-vis payers. And in demographics where travel distance is long, that every 3-week dosing can matter to some patients.
So is that the deciding factor? Is there cost differences between the agents that we know?
I'm not aware of any real cost difference between these agents. My understanding was that they all come in at roughly 12 grand a month. Not an economist to this stuff, but something like that. Regardless of which you use. But to the patients, it can matter. I mean, I hand out a lot of gas cards via our local advocacy organization because people have trouble affording the gas to come into my cancer center. And in that context, with that kind of population, people traveling either because they can or because they have to, it matters a lot.
So you very nicely summarized the 3 drugs in the second line space. Obviously, only one of them is dependent on PD-L1 status. How does the panel feel about the use of PD-L1? I know it's kind of artificial now because we've all agreed we should check it in the first line setting. Prior to that, we didn't necessarily do this to administer second-line therapy. In fact, most people didn't do it. We're going to come back and circle around to the keynote 21G issue in a moment, but what's the message about if you're PD-L1 negative in the second-line space? Does that dissuade anybody from using these agents, or do you see, since two of them are agnostic to PD-L1, as their standard of care?
But it's not just the approval. It's that the PD-L1 negative patients, as measured the way we're currently measuring them, did receive benefit in the now 3 major efforts evaluating them.
I think the key thing is, what is your alternative? So if our alternative is single agent docetaxel, which is not a great drug, then any of these drugs, even though the benefit may be relatively low in those who have low PD-L1 expression, it's still better than the alternative. Now, if we get a better cytotoxic sitting in there or something else, that becomes up for debate again.